Thank you for that introduction. The main point of my presentation is to describe the process of evaluating abuse-deterrent properties of opioid drug products that are designed to make abuse through the nasal route difficult. To do this, I will describe tablet manipulation method and design of a clinical PK study that we conducted to evaluate the extent of manipulation needed to defeat control release properties of an abuse-deterrent formulation when milled and insufflated. This study was designed and conducted based on recommendations made in the FDA Guidance for Industry General Principles for evaluating the abuse deterrent of generic solid oral opioid drug products. The evaluation process is a two-stage process. In the first stage, we need to see if we can manipulate the drug product into particle size ranges that are easy to insufflate, and if so, characterize the manipulated product. And once that's done, in the second stage, we'll do a pharmacokinetic evaluation of the manipulated drug product by conducting in vivo PK studies. These studies should be conducted in recreational opioid users that are otherwise healthy as per the guidance. In the next few slides, I'll describe the study objective and study design. In this study, we are evaluating a polyethylene oxide-based oxycodone tablet formulation with nasal abuse-deterrent properties by comparing it to an immediate release formulation. We also will use two different formulations of polyethylene oxide-based oxycodone abuse-deterrent formulations to evaluate the effect of polymer to active ingredient ratio on nasal bioavailability of manipulated abuse-deterrent formulations. One of these formulations has a ratio of 3.9 and the other has a ratio of 2.1. And of course, we will evaluate the adverse events that are observed during this study. We have two abuse-deterrent formulations in this study. One is the one with the polyethylene oxide to API ratio of 3.9. We manipulate this to two different particle size ranges, 106 to 500 micrometers and 500 to 1000 micrometers. And these are treatments A and B. We have treatment C, which is the second abuse-deterrent polyethylene oxide formulation, and that has a ratio of polyethylene oxide to active ingredient of 2.1. This tablet is manipulated to particle size range of 106 to 500 micrometers. And finally, we have the control formulation, which is an immediate release formulation of oxycodone tablets, which is milled to a particle size range of 106 to 500 micrometers. The study is a single center randomized open label single dose 4 sequence 4 period 4 treatment crossover design under fasting condition in 41 healthy recreational opioid users. In this study, we're comparing formulation A versus B to evaluate the effect of particle size on bioavailability, and formulation A versus C to evaluate the effect of difference in polymer to active ingredient ratio on bioavailability. This slide shows an example of subject dosing. The confinement period is 12 days. Subjects are dosed on days 1, 4, 7, and 10, and the washout period is 72 hours between doses. That's an adequate washout period given oxycodone half-life. To put the objectives of the study in simpler terms, we want to see what level of manipulation is required to defeat abuse-deterrent properties of abuse-deterrent oxycodone ER tablets for nasal route. To do this, we take oxycodone oxy-ER1 tablets and mill that to two different particle size ranges 106 to 500 micrometers, and 500 to 1000 micrometers. And compare that with a milled and insufflated OxyIR tablet, which is also milled to 106 to 500 micrometers. And in this study, we've used naltrexone block to block the euphoric effects and other adverse events of oxycodone. As for the second objective of the study, we're comparing two different formulations with two different polymer to active ingredient ratios. We take oxycodone ER, 1 tablet mill dose, particle size range of 160 to 500 meters and micrometers, and Oxy ER 2 tablets, and we mill dose to particle size ranges of 106 to 500 micrometers. 41 subjects were enrolled in this study. All 41 were included in safety populations. 36 subjects were included in PK analysis, and 33 subjects completed the study. Over the next few slides, I'll describe the process of tablet manipulation and study dose preparation. The first step in manipulation process is to see whether we can manipulate the tablet into a particle size range that can be insufflated, which we successfully did in our study. And once we manipulated the tablets into a particle size range that can be insufflated, then the resulting powder was characterized by looking at particle size characteristics, drug loss during manipulation, and also drug load of the resulting powder. This slide shows you a picture of the milling equipment that we used, and also pictures of the 4 treatments that were prepared, the manipulated products. And the table on the top, what I want to draw your attention to is the fact that treatments A and B were the same abuse-deterrent tablet, milled to different particle size ranges. But you see that we needed 266 milligram of the finely milled powder to get a dose of 30 milligram of oxycodone versus 252 milligram of the coarsely milled powder to get a dose of 30 milligram of oxycodone. So there is a differential drug loss in the manipulation process. This slide shows you the particle size distribution between the different treatments. The slide on the left, the figures on the left show the particle size range distribution for the adverse abuse-deterrent formulations. We will see there that the bulk of the particles are in the range of 300 to 500 micrometers, whereas for both Oxy ER1 and Oxy ER2 tablets, whereas the bottom right, the particle size range is within 100 to 300 micrometers for the immediate release formulation. And top right is the particle size distribution for the coarsely milled oxycodone tablets, and there the bulk of the particles are in the range of 500 to 850 micrometers. This slide shows the recovery of powder and the recovery of drug in the process of manipulation. The figure on the left, the gray bars, show the recovery of powder, which across all treatments we see that we have nearly recovered 100% of the mass of the tablet as powder. And the blue bars show recovery of the drug. And for the immediate release product, the recovery of drug is also nearly complete. However, for the abuse-deterrent formulations, the recovery of drug ranges anywhere from 45% to 70%, depending on the formulation and particle size range. And the figure on the side shows the polymer to active ingredient ratio in both the intact tablet and the manipulated product. The gray bars show the intact tablet, and the blue bars show the ratio for the manipulated product. As you see, the ratio increases once the product is manipulated into powder, again indicating the drug loss during the manipulation process. The slide shows the preparation of the study doses. Top left, tablets are milled in bulk, 160 tablets per batch. Once that's done, they're transferred into vials that are intended for shipment to the study site, each vial containing 8 to 9 doses. And once they are at the study site at the pharmacy prior to administration of the drug, the pharmacist transfers equivalent of 30 milligram dose of oxycodone into each dosing vial, which is then sent to the floor for administration to the subjects. And the table on top right shows that the drug load for treatments for the 4 different cohorts, the study was conducted in 4 different cohorts due to logistic reasons, was fairly consistent, resulting into the same amount of powder more or less be administered for each dose in each group. Now for the pharmacokinetic evaluation result. This is the time concentration profile for all 4 treatments in this study. As you can see, the Cmax and AUCs for the IR product and the 2 abuse-deterrent products milled to fine particle size are fairly close and grouped together on top, and the Cmax for the coarsely milled oxycodone ER product is considerably lower than the other 3. Here we have the comparison between the 2 abuse-deterrent formulations, finely milled, and the IR product. The point estimates for Cmax partial AUC, AUC 0 to T, and AUC 0 to infinity were all within the 80 to 120% bioequivalence limits. I apologize, that's supposed to be 125%. 80 to 125%. 120 is a typographical error. And the upper limit of confidence intervals for our measured parameters was above 80. Recall that here where the control is the reference and the 2 abuse-deterrent formulations are the test product. In this figure, in this table, we're comparing the coarsely milled oxycodone product, abuse-deterrent oxycodone product with the immediate release product. The upper limit of confidence interval for all 3 parameters was below 80%, and the point estimate and 90% confidence intervals of AUC 0 to T and AUC 0 to infinity were within 80 to 125%. Now, this is showing the PK parameter values for the comparison of the coarsely milled oxycodone tablets to the immediate release product. As you can see, the upper limit of 90% confidence interval for Cmax, AUC 0 to 0.5 hours, and AUC 0 to 4 hours all fall below the 80% bioequivalence level, indicating that this drug, at least up to 4 hours, clearly has lower bioavailability than the reference, the immediate release products. If you look at the AUC 0 to T and AUC 0 to infinity, you will see that overall it has the same bioavailability as the reference product. However, our focus here really is up to 4 hours. Cmax, AUC 0 to 0.5, and AUC 0 to 4, because this is the time period which matters in terms of the drug having its euphoric effect and therefore inclines people to abuse it. Here, similar to comparing the immediate release formulation with the coarsely milled formulation, we're comparing the finely milled formulation, which we already showed that it's more or less behaving similar like the immediate release formulation with the coarsely milled oxycodone tablets. Here we're using the coarsely milled as the reference product. So we're looking at the lower limit of confidence intervals, and we'll see that the lower limit of confidence intervals for Cmax, AUC 0 to 0.5 and AUC 0 to 0.4, 0 to 4 hours all fall above the 125% bioequivalence limits, indicating that the bioavailability of the coarsely milled oxycodone hydrochloride tablets is considerably below the bioavailability of finely milled oxycodone ER tablets for the time range that we're interested in as far as abuse deterrence is concerned. This slide shows the comparison of treatment C versus treatment A. The pharmacokinetic parameters show that there is really no difference between the two treatments in terms of bioavailability. The confidence intervals for all PK parameters are within the 80 to 125% bioequivalence range. So in conclusion, the coarsely milled abuse-deterrent oxycodone ER tablet had lower nasal bioavailability than immediate release oxycodone tablets. Finely milled abuse-deterrent oxycodone ER tablets did not demonstrate lower nasal bioavailability than oxycodone IR tablets. At particle size range of 106 to 500 micrometers, differences in polyethylene oxide to oxycodone ratio did not seem to affect nasal bioavailability. A total of 114 treatment emergent adverse events were reported in this study. 102 of those were deemed to be related or possibly related to oxycodone. 5 subjects withdrew from the study due to mild treatment emergent adverse effects that were considered expected and at least possibly related to study drug. 5 subjects required co-administration of concomitant drugs to treat treatment emergent adverse effects, the majority of which were expected and considered at least possibly related to the study drug. Now I have a couple of challenge questions for you. Challenge question 1. What's the objective of nasal PK study in the context of ANDAS? A. To establish that the test product is abuse deterrent. B. To establish that the reference product is abuse deterrent. C. To establish that the test product is no less abuse deterrent than the reference product. D. To establish that the reference product is no less abuse deterrent than the test product. The correct answer is C. To establish that the test product is no less abuse deterrent than the reference product. Challenge question 2. Which of the following statements is not true? A. The study showed that PEO to API ratio influences nasal bioavailability of oxycodone. B. No significant differences were observed in AUC 0 to infinity of coarsely milled oxycodone AD tablets and AUC 0 to infinity of milled oxycodone IR tablets. C. Coarsely milled oxycodone AD tablets have lower AUC 0 to infinity, AUC 0 to 4 than finely milled oxycodone AD tablets. And D. No significant differences were observed in the bioavailability of finely milled oxycodone AD tablets and milled oxycodone IR tablets. The correct answer is A. The study showed that the polymer to active ingredient ratio influences nasal bioavailability of oxycodone. This is not a true statement. As you remember, we showed that the ratio did not matter in terms of bioavailability. So the overall take-home message, oxycodone ER tablet multi-particle size range of 500 to 1000 micrometer retain ER property while milling the tablet to particle size range of 106 to 500 micrometer resulted in loss of ER property. Polymer to oxycodone ratio at the particle size range of 106 to 500 micrometer does not affect PK parameters. Manipulation of oxycodone ER tablets resulted in considerable oxycodone loss. That concludes my presentation. I would like to acknowledge the FDA team that designed and oversaw the conduct of this study and the data analysis, the Wins and Associate team that conducted the clinical study and bioanalysis, and the Purdue team that designed and conducted the in vitro studies and prepared the study drugs.
The second presentation of this session is also related to abuse-deterrent formulations of opioids, but the focus will be on the consideration in assessing the in vitro studies. The title of this presentation is Advancement in the In Vitro Evaluation of Abuse-Deterrent Formulations for Opioid Analgesics, Research and Assessment Perspectives. I am Manar Al-Ghabeish, and I will be pleased to guide you through this presentation. I'm from Office of Pharmaceutical Quality, Office of Testing and Research. What is pharmaceutical quality? A quality product of any kind constantly meets the expectations of the user. Drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is constantly meeting standards that ensure every dose is safe and effective, free of contamination and defects. It is what gives patients confidence in their next dose of medicine, and that includes abuse-deterrent formulation. The objective of these presentations are to describe FDA research effort in evaluating abuse-deterrent formulations, provide key considerations in assessment of in vitro abuse-deterrent studies, and provide example of common deficiency in in vitro methods. A brief history behind the U.S. opioid epidemic. In the early 2000s, reports of overdose and death from prescription pain drugs, especially opioids, began to rise sharply. In 2017, U.S. Department of Health and Human Services declared the opioid crisis a national public health emergency. 2019 statistics showed that more than 10 million Americans misuse prescription opioids, and more than 70,000 Americans died from drug overdose. Opioids are claiming lives at a staggering rate, and overdose from prescription opioids are reducing the life expectancy in the United States. That's why effort in addressing the crisis of abuse of opioids, while still providing patients in pain access to effective relief, is a top priority of the FDA. One of the areas FDA efforts has focused on is supporting cutting-edge research. And that includes help in expediting the development of a more effective abuse-deterrent formulation of opioid drugs and non-opioid alternative for the treatment of pain. But what is abuse-deterrent formulations, ADF? ADFs are formulations that have the potential to make abuse of these products more difficult or less rewarding. They are meant to deter abuse and not necessarily prevent abuse. Abuse deterrence, AD, can be achieved using several designs, such as the use of physical or chemical barrier. A common example is the use of a polymer such as polyethylene oxide, PEO, that makes the drug product hard to crush and makes the extraction of the drug difficult. Another design is the use of agonist and antagonist, where the antagonist of opioids, such as naloxone and naltrexone, is clinically inactive if the drug product administered orally. However, if the product is crushed and injected or snorted, the antagonist becomes active and reduces the effectiveness of the opioid and deter abuse. Last example is aversion, which is the use of a substance that produces an unpleasant effect if the drug product is manipulated. An example of aversion is including a substance that causes irritation to the nasal mucosa if the drug product is crushed and snorted. Current regulatory consideration in ADF. There are two general guidances for ADF. One is guidance for industry, abuse-deterrent opioids, evaluation and labeling, published in 2015. This guidance is for new drugs. However, it states the basis for AD studies, including the in vitro or category 1 studies. The second one, which is for generic ADF, is general principles for evaluating the abuse deterrence of generic solid oral opioid drug products, which is published in 2017. In addition, there are 8 product-specific guidance, PSGs, that include the recommendation not only for bioequivalence study, but also for in vivo and in vitro AD studies. You can visit this webpage on the left for information and updates from the agency about ADF. This table lists the opioid product with FDA-approved AD labeling. The AD description can be found in section 9.2 of the label. I added the links for the labels for the 10 brands, so you can check the description of AD designs, studies, and routes that these products intended to deter the abuse from. Also, I added the links for the current PSGs for these products. There is only one approved generic, and hoping soon we'll see more generics. The general guidances and PSGs and the approval of ADF are the products of the knowledge gained from assessing NDAs and ANDAs, knowledge from testing on ADF under assessment by the method verification program of FDA, external research collaboration with institutes like NIPT, and our own internal research. Before discussing the internal research, let's go through a first challenge question. Which of the following is not an abuse-deterrent design? Pill bottle lock, aversion, physical barrier. The correct answer is pill bottle lock. Even though it can help in limiting the access to opioids to non-patients, but it's not a formulation design. Internal research of abuse-deterrent formulation. ADF internal research included several areas, which are reference-listed drug RLD characterization, surrogate AD properties and shelf life, new techniques and procedure, standardization of methods, material and process impact on AD properties, threshold identification, safety of ADF, after-market risk-benefit assessment, and naloxone product quality. I will be talking about some of the examples of the research done in some of these areas. The first example is the impact of materials and process on the AD properties. The design feature of an ADF is a result of manufacturing process, material attributes, or a combination of both. That's why it's crucial to understand the relationship between the process or material and the AD properties. Some of our work that has been already published investigated the manufacturing process of ADF, such as the direct compression of tablets containing PAO of various grades and percentages, and the subsequent curing step. Other work involved the hot melt extrusion of PAO containing powder mixture. Both studies showed the impact of the process parameters and the PAO as an AD polymer on the AD properties evaluated, like the hardness profile. Another example is the surrogate AD property for syringeability and injectability. Viscosity is a key material attribute affecting syringeability and injectability. In this study, we measured the withdrawn volume and the injectable force by texture analyzer and used them as indicator for syringeability and injectability of PAO mixture. The PAO of low molecular weight and thus low viscosity showed a significant higher withdrawn volume and a lower injectable force compared to the PO of higher molecular weight. This PO probably then may not be suitable to make a formulation which is abuse-deterrent by syringeability. The last example is the standardization of method. In this example, I will be talking about milling as a physical manipulation method. Physical manipulation is critical and the initial step for most of in vitro and in vivo AD evaluation study. Manipulation condition and thus the efficiency can impact the following AD evaluation. In this study, milling conditions like number of tablets used, milling time, drug loading were tested and found to impact the recovery and particle size, especially the fine particles %. And here I listed selected publications which have been presented in many conferences. Moving to talk about the consideration taken by the agency in assessing generic ADF, specifically the in vitro studies. These considerations can also be used by generics while evaluating AD properties of the proposed drug products and designing the in vitro method. I will be highlighting the logic and the research behind these considerations. The first point to consider in assessing an ADF is what are the product quality attributes? Generic drug product is not required to be Q1, Q2, meaning the inactive ingredients do not need to be qualitatively and quantitatively same as that used by the RLD. And the AD evaluation is a performance-based. However, it is important to do a risk analysis and identify what are the product quality attributes that is related to the AD properties, including what is the AD design, is the material manufacturing process or both responsible for the AD design, because inadequate attributes may lead to failing the AD evaluation. An example for this risk analysis is if the RLD has a polymer that makes the product hard to crush, does the proposed generic also has an excipient that will make the product hard to crush? The second point is strength to be evaluated. The drug product may have several strengths. When evaluating the drug product for AD properties, all strength can be evaluated, or the applicant can evaluate selected strength if an acceptable justification is provided. For the same example, the applicant can provide the ratio of opioid to polymer responsible for the AD properties for all the strength, and then pick the strength that cover the range of ratio, the maximum, minimum, and middle for the evaluation. The third point is evaluating all potential routes of abuse. Looking at the label for the RLD, the current RLDs have an approved AD label for 1 to 3 routes of abuse. However, it is expected that the generic drug to be no less abuse deterrent than the RLD with respect to all 4 routes of abuse. And that's to ensure that the generic drug will not exhibit any more risk than the RLD. After those initial points, looking at the general scheme of comparative in vitro studies that can also be found in the generic guidance, the AD evaluation starts with 2 comparative studies between the RLD, R, and the proposed generic product, T. The 2 studies are physical manipulation and extractability. The physical manipulation and extractability help in the subsequent comparative evaluation of R and T with respect to the 4 routes of abuse: oral, parenteral, nasal, and inhalation. To complete the evaluation, the applicants conduct other in vitro studies such as syringeability for parenteral route and smoking test for inhalation route. And when necessary, the applicant also conducts clinical studies such as oral and nasal pharmacokinetic studies. Another key consideration in designing and selecting AD method and condition is to make them risk-based. The benefit of that is to reduce burden on testing on the applicant and burden on assessing them without weakening the effectiveness of the evaluation. For example, the applicant may perform physical manipulation on CNR using many tools and conditions, followed with extractability of all the manipulated product in many extractability conditions, such as using several solvents and at different temperature. That would result in thousands and thousands of tests and massive data. On the other hand, based on abuse risk, the applicant can select at least 2 extreme manipulation forms of the drug product, which are the intact product and the most effective manipulated form, and use them for the subsequent studies. Also, the subsequent studies like extractability can be performed on a tier-based approach, in which R and T are tested using the same condition in one tier, and if they didn't fail, then the applicant can move to a tier with a more complex or difficult conditions. Moving to examples of actual deficiency that show how the internal research helped in the assessment. The first examples are related to the physical manipulation. From internal research, we found that the subsequent extractability and extraction percent can be an underestimation of the real values due to drug loss during manipulation, and that the drug loss is highly dependent on the number of the units used during the manipulation process. This information has been communicated to applicants in deficiency and also is in the product-specific guide. Another example related to manipulation and the selection of manipulated form for the subsequent studies. In this specific example, the product design and structure are responsible for the AD property. So the release of the drug from the intact product, ROT, during extractability was so low, less than 10%, which made it very hard to make a meaningful comparison. On the other hand, the most effective manipulation destroyed the structure and the AD properties were lost, causing very high release, like more than 50%, so that even R failed. That's why introducing an intermediate condition that retains some of the structure could be more suitable in the comparative studies. The last examples of deficiency are related to statistical analysis, and those are one of the very common deficiencies that were communicated to applicants in the early submissions, especially before the general guidance. The applicant would run many tests using 1 or 2 units that was not enough to conclude that C is no less abuse-deterrent than R. This is why sample size and number of replicates, along with non-inferiority tests, will support the conclusion of the comparative studies. Before concluding my presentation, here is another challenge question. Which statistical test should be used to compare T with R? Paired t-test, non-inferiority test, regression analysis. The correct answer is non-inferiority test, which is used to compare 2 independent variables. The summary for this presentation is FDA has conducted comprehensive research in crucial areas of ADF. ADF research has supported the assessment of in vitro AD studies. Common deficiency in the AD in vitro evaluation can be avoided with well-designed method. And my last message is AD properties can be defeated with varying degree of difficulty, and hence iterative improvement on the existing AD technology and more innovative designs are really needed. Thank you for listening. If you have any question, I will be glad to answer them during the panel discussion.
Hello everyone. The topic for my presentation today is PBPK absorption modeling and virtual bioequivalence to support generic drug development and regulatory decision-making for oral products. Here is my disclaimer. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. Through this presentation, the participants will understand PBPK absorption modeling and questions that can help assist generic drug development. The participants can also learn case examples on developing PBPK modeling and conducting virtual BE simulations for supporting regulatory decision-making. In this presentation, we will first introduce the applications of PBPK absorption model and the highlights of PBPK impacts in Office of Generic Drugs. Then we will introduce the guidances related to PBPK, followed by recent regulatory review and research examples. Lastly, we will talk about the challenges and opportunities related to PBPK absorption models. Currently, new drug applications containing PBPK absorption modeling and simulation related to biopharmaceutical assessment is evolving. The applications covered aspects including setting clinically relevant dissolution specifications, setting critical material attribute specifications, risk assessment, pH-dependent drug interaction potential prediction, food effect, and biowaiver. As shown in the left figure, the number of new drug applications containing PBPK absorption modeling and simulations for biopharmaceutical assessment is increasing up to the year 2015. From 2015 to 2018, it seems that there is a reduced number of NDA submissions, which could be due to the reason that some submissions with PBPK modeling were included in the IND, investigational new drugs, and the IND numbers were not included in the summary. The right figure shows that there were more applications containing PBPK absorption modeling in the area of setting dissolution specifications, setting particle size distribution specifications, and risk assessment. This slide shows the regulatory questions that PBPK absorption model can answer in generic drug development. They are all related to BE assessment, including setting dissolution safe space, evaluating the impact of changes in critical quality attributes, assessing the risk of formulation, mechanism changes, etc. The PBPK absorption modeling can also be used to predict PPI-DDI potential and the GI local concentration, support BE assessment in specific populations, and the waiver of in vivo studies. This figure shows an overview of PBPK absorption modeling and simulation in ANDA submission up to 2018. We can see that major objective is to support dissolution specification settings. The second often objective is to support bioequivalence assessment. This table summarizes the highlighted cases where research of PBPK absorption modeling had impacts on regulatory decision-making. For example, PBPK was used for conducting risk assessment of not conducting in vivo studies for lower strength of oxybutynin generic products when bioequivalence has been established at higher strengths and the dissolution has deviations. PBPK was also used to support that less than 20% free base in procedural hydrochloride product can ensure an in vivo BE of the generic product. PBPK can also be used for supporting the BE study design, for example, determining the PK matrix for BE evaluation for mesalamine suppositories and evaluate whether a two-way crossover study in combination with PBPK modeling can meet the guidance recommendation of a four-way crossover study for BE assessment for methylphenidate product. The other cases include using PBPK for the risk assessment of a release mechanism change, PPI impact, and alcohol dose dumping. In the year of 2020, FDA issued two guidances supported by PBPK regulatory applications and research. One guidance is the use of PBPK analysis, biopharmaceuticals applications for oral drug product development, manufacturing changes, and controls. Another guidance is evaluation of gastric pH-dependent drug interactions with acid-reducing agents. These two guidances describe the utility of PBPK absorption modeling for biopharmaceutical applications as well as for predicting the pH-dependent DDI potential. This figure shows the general PBPK modeling procedure in ANDA submissions. Generally, the model input include in vitro data, for example, dissolution data of both test formulation and reference formulation, as well as available clinical datasets. Then use IV and oral in vivo data to estimate disposition and absorption parameters. After verification validation of the model, sensitivity analysis could be conducted to identify the key factors that impact the in vivo performance. The model can then be used to predict in vivo PK of different batches. Virtual BE simulations can also be conducted to set dissolution safe space or to simulate BE trials between reference and test products. In the next couple of slides, I will show a few examples of using PBPK absorption models in the regulatory work. The case example shown here is about a capsule product for which the PK parameters were found to be sensitive to changes in mean particle size of drug X under fasting condition. The applicant submitted a mechanistic absorption model to link particle size distribution with in vivo PK data. FDA reviewers conducted assessment on the PBPK modeling and simulations, which suggested that with a certain fixed value of D50 for all strengths, the test versus reference PK matrix showed a low risk of non-BE when D90 changed over a wide range. These modeling results suggest a satisfactory BE assessment of this ANDA and setting a clinically relevant 3-tier PSD specification. This case example is about using PBPK absorption modeling to support waiver for lower strengths with in vitro testing deviations. We know that the waiver of lower strengths can be dependent on formulation proportionality, dissolution similarity, bioequivalence on other strengths. However, this case example has dissimilar dissolution profiles for lower strength of the test product. Thus, PBPK-PBPM modeling was used for predicting the impact of faster release of lower strength on the bioequivalence under fasting and fat conditions. After review, we identified some deficiencies with the submitted model, especially on the validations. When these deficiencies are addressed, the developed PBPK-PBPM model can be used in assessing the impact of dissolution differences on in vivo performance bioequivalence. The case example shown in this slide was based on the regulatory research. According to FDA draft guidance for BE studies with PK endpoint for drugs submitted under an ANDA, generally both fasting and fat in vivo BE study are recommended for immediate release product, unless the product should be taken only on an empty stomach. However, normally only fasting study is recommended for BE evaluations for other agencies. For harmonization, can we identify some certain situations that only fasting studies can be recommended for the establishment of BE? As shown in the right figure, we established and validated a PBPK modeling to predict the drug PK under both fasting and fat conditions. This demonstrates the potential utility of PBPK modeling to assess the risk of bioinequivalence attributable to food intake. Another area that PBPK can be used is for supporting biowaiver. Currently, there are two main general guidances for BAB waivers based on BCS. One is the guidance for waiver of in vivo BAB studies for IR oral products based on BCS, which was issued in December 2017. The other one is ICH-M9 guideline for BCS-based biowaivers, which was endorsed in June 2018. Both guidelines describe that For BCS Class 3 drug products, Biowaiver should be supported by high solubility of the drug substance, rapidly dissolving of drug product, and the test product formulation is qualitatively the same and quantitatively very similar. Here is the question, whether and how we can expand BCS Class 3 Biowaiver to non-Q1, Q2 product. We have internally developed a PBPK model for putative BCS Class 1, 3 drug, oseltamivir phosphate, OP, and its metabolite, oseltamivir carboxylate, OC, in both adults and pediatrics, then conduct virtual BE simulations to establish BE dissolution safe space for OP in both adults and pediatrics. This case example not only highlights the utility of PBPK absorption modeling and simulation for setting clinically relevant dissolution specifications in both adults and pediatrics populations, but also demonstrates the potential utility of PBPK modeling as alternative BE approach to support biowaiver of non-Q1, Q2 BCS Class 3 drugs. Details can be found in this AAPS journal paper listed here. When using PBPK absorption model, there are always some challenges but also opportunities for addressing those issues. For example, more understanding is on the upper part of GI tract, and we need further understanding of physiology in the lower part of the GI tract, such as colon for the drugs which will be absorbed in this section. We also need consistent and adequate approach of generating and incorporating biorelevant solubility, dissolution profiles, QC or biorelevant, permeability and particle size change as model inputs. In addition, when building or optimizing the PBPK model, those uncertain drug formulation-dependent or system-dependent parameters will need adequate justifications. Most importantly, purpose-dependent model verification validations are needed. To resolve the challenges and fill the knowledge gap, OGD has funded a couple of contracts and grants in this area. For example, the contract Better Understanding Risk Mitigation in the Evaluation of Relative Bioavailability of Pediatric Generic Products is to evaluate risk factors associated with the BE assessment for pediatric generic products and whether biorelevant dissolution and PBPK modeling can help mitigate risk. The contract expanding BCS Class 3 waivers for generic drugs to non-Q1, Q2 products is to assess the impact of excipients on the dissolution and permeation of BCS Class 3 model drugs in solid oral dosage forms. The research results could increase the confidence of using certain amount of some excipients. In addition, to investigate the impact of excipients on the in vivo performance of BCS Class 3 drugs, PBPK modeling may potentially be used as alternative BE approach and to support biowaiver of non-Q1, Q2 BCS Class 3 generic drugs. Another grant was just recently awarded to investigate PBPK models of oral absorption to simulate BE studies. As demonstrated above, we have conducted research related to oral PBPK modeling through both internal and external projects. Here listed a couple of recent publications based on these research projects. You may refer to these publications for details. In conclusion, these cases demonstrated that PBPK absorption modeling and simulation have been increasingly used in generic drug applications. With continuous improvement along with more submissions and research, PBPK absorption modeling and simulation would aid in setting clinically relevant specifications, conducting risk assessment on the impact of critical quality attributes, evaluating the impact of excipients and food on bioequivalence, supporting high-impact applications such as alternative BE approach and biowaiver. FDA also encouraged the sponsor to submit alternative BE approach proposals with modeling and simulation data, communicating with agency at early stage via controlled correspondences or pre-AND meetings is encouraged. Now, through this presentation, we have learned some applications of PBPK absorption modeling for oral products. Here, I have one challenge question for the participants to think about and answer. PBPK absorption modeling can be used for A. Assessing the impact of particle size distribution on BE. B. Supporting BCS Class 3 drug waiver. C. Risk assessment of bioinequivalence attributable to food intake. B. All of the above. The answer is all of the above. With that, I would like to thank all the people listed here for their support and hard work, especially the reviewers and scientific assessors involved in the regulatory work. Also, special thanks to those authors who are involved in the publications that I mentioned in the slides. This concludes my presentation, and I will be back later to answer questions during the panel discussion. Thank you.
Good afternoon, everyone. I'm Dr. Melanie Mueller. I'm a lead toxicologist in the Office of Generic Drugs, and today I will be presenting PharmTox safety review of flavor additives in generic drug products that are intended for use in humans. I'd like to start off by showing the learning objectives we're going to cover today. We will begin with defining and describing flavors in drug products and also identifying the regulatory framework. Next, we'll dive into the CDER PharmTox safety assessment of flavors and its challenges. We'll learn about the principles and approaches taken during our reviews and also the steps we take to navigate complexities that are associated with flavor reviews. And last, we will review some case studies that demonstrate how PharmTox approaches safety flavor review and how we navigate complexities that are commonly encountered during our reviews. Okay, before we talk about flavors, I would first like to give you a few general words about how PharmTox reviews generic drugs. In OGD, PharmTox conducts a comparative safety assessment on a consult basis. During our assessments, we ask questions such as whether differences between the reference listed drug and generic drug impacts the safety of the formulation, and whether the safety profile of the generic drug is the same as that of the reference listed drug. We conduct a risk-benefit assessment considering the specific context of use of each drug product under review. And during our reviews, we employ principles and approaches that are similar to those used by our PharmTox colleagues in the Office of New Drugs. OGD PharmTox is consulted yearly on approximately 10% of the ANDAs, and roughly 25% of these consults are for excipient-related issues, which include flavor additives. Because generics often lack extensive safety data, the excipients are typically assessed from both non-clinical and clinical perspectives. And during our assessment, the context of use is key, and our assessment includes a risk assessment in vulnerable patient populations, such as neonates, pregnant women, and patients with renal failure. And with that, we're ready to dive into the details of safety review of flavor additives. Flavor additives are excipients and are reviewed as such. Excipients are defined as ingredients that are intentionally added to therapeutic and diagnostic products. Flavor additives are added to increase palatability of oral formulations. Oral formulations might include pediatric formulations, gums, lozenges, and orally disintegrating tablets. Excipients themselves should not exert a therapeutic effect at the intended dosage. And excipient safety needs to be demonstrated before a pharmaceutical can be marketed. Flavor additives, such as all excipients, are regulated by the Federal Register. 21 CFR regulation sets the standard for flavor additives. Regulations by CFR are laws and must be followed. FDA guidance present FDA's best thinking on how to apply regulatory recommendations, and guiding documents as such are not binding. However, they help us to apply the regulatory framework to our reviews. And here you see a list of FDA guidances that we commonly apply during our safety review of flavor additives in generics. And with that, we arrived at our next learning objective, on how PharmTax conducts safety flavor review and how we navigate challenges that we commonly face during review of flavor additives in generics. To start our second learning objective, I would like to reiterate the PharmTax principle underlying our reviews. We conduct a risk-benefit assessment considering the specific context of use of a drug product. Hereby we consider the dose, the duration of use, the patient population, as well as the route of administration. In new drugs, flavor additives often are qualified during drug product development. It means that clinical and non-clinical data that also include the flavor are submitted for review. In generics, on the other hand, flavor safety often relies on prior findings of clinical safety. Okay, but what do I mean by that? Applying prior findings of clinical safety during our review. Well, for one, we look to the IID, the Inactive Ingredient Database. If we can identify FDA-approved products that contain higher levels of the proposed flavor or the individual flavor components, then no further justification is needed and the flavor is qualified for safety. However, keep in mind context of use matters, so not all products listed on the IRD that contain the same flavor are suitable to adequately qualify the flavor in a proposed product. Secondly, we review published human and non-clinical safety data to justify the flavor safety. All information needs to support that the generic has a similar safety profile as that of the reference listed drug. There's also an avenue for applicants to get pre-submission guidance on formulation. Such guidance can be requested via controlled correspondences. Next, I'd like to talk a little bit about two different approaches we might take during our flavor safety assessments. The first approach is that we would assess the safety of a flavor as a whole. The second approach is that we assess the safety of the individual components of a flavor additive. The review approach typically is determined by either the nature of the submitted data or whether there are prior findings of safety available for the exact proposed flavor additive. However, regardless of the approach, the same review principles, as I already mentioned earlier, apply, which are a risk-benefit assessment under a specific context of use. Now, I'm going to talk about the challenges we encounter during our flavor safety assessments. This slide lists the challenges in a nutshell, but over the following few minutes, I'm going to dive a little deeper into underlying causes of these challenges and the steps we take to navigate the same. A big challenge we encounter during our reviews is due to the complex nature of the flavor additives. Depicted here is the composition of a pineapple flavor. As you can see, this pineapple flavor is a rather complex mixture that is composed of approximately 30 individual components. Now picture that we have to review the individual components of a flavor rather than the flavor as a whole. And you can imagine that our review process becomes rather cumbersome and time-consuming. However, we have an approach that we apply to alleviate our review burden. We apply safety thresholds that are founded in PharmTax principles, considering the context of use of the drug product in which the flavor additive is present in. Now, applying these safety thresholds allows us to limit review to those flavor components for which the maximum daily intake exceeds the safety thresholds that we determine to be appropriate based on the context of use of the drug product. To recap, flavor additives are complex mixtures which can lead to a cumbersome review process when assessing individual components of a flavor agent. But you also heard how PharmTax applies safety thresholds based on the context of use of the drug product to enhance review efficiency. Yet there is another challenge that is caused by the complexity of these flavors. And that is because no two flavors are alike. What that means is you could have flavors with the same name, such as pineapple flavor and pineapple flavor, yet their composition is qualitatively and quantitatively completely different. This often leads to inadequate reference of the inactive ingredient database when an applicant submits their generic product. However, to better guide industry, PharmTax drafted language for FDA guidance that describes expectations for a successful ANDA submission. Shown here is the composition of two different pineapple flavors. These examples demonstrate how flavors with the same taste profile can differ greatly in terms of their qualitative composition. As I mentioned, such differences in composition often lead to inadequate references to the FDA inactive ingredient database upon generic submission. That is because the generic applicant does not know the composition of the FDA-approved flavor. And that brings me to the next big challenge we very often encounter during safety review of flavor additives. During our flavor safety assessment, we commonly encounter internal and external challenges because of the proprietary nature of flavor additives. Usually, the composition of flavors is proprietary because they're commonly made by third-party companies. Internally, that's causing sometimes a delay in review because the quantitative breakdown of a flavor is often not readily available for our review, and we need to request it during the review. However, if present, the information may be submitted to FDA under a drug master file. Externally, however, applicants are also not always privy to proprietary information under a DMF. That could lead to issues in which, for example, unknown to the applicant, flavor additives may contain components that are not safe for the intended patient population. Or applicants may not know of changes in the flavor composition. And for us, that means we need to exercise caution when communicating with the applicant to not reveal proprietary information of a flavor. To alleviate these problems, PharmTax drafted language for FDA guidance that specifies that the quantitative breakdown needs to be readily accessible for FDA to conduct a comprehensive review of the safety of a flavor. The third item on my list of challenges are the criteria used to flag safety issues. What that means is that sometimes flavor manufacturers provide statements of safety, which are then included in the generic or into a new product. Such statement could be something along the lines of, our flavor is safe for use in your product because all components are either GRAS or they do not exceed 0.1% of the total flavor volume. However, these approaches do not consider the context of use of a drug product. For example, the maximum daily exposure to a component present at 0.1% in a flavor that, however, is in a drug product with a large maximum daily dose quickly may exceed the safety thresholds we apply during our reviews. Second, while GRAS is safe for oral exposure via diet, exposure levels in the clinical population are not considered with a GRAS statement. For example, propylene glycol is listed as GRAS, but it presents safety signals in the pediatric patient population. To reiterate, during our assessment, PharmTax considers the context of use and the maximum daily exposure to flavor or flavor components, just like we do for any other excipient that we evaluate. And that means that the amount of a flavor as a percentage of the total formulation is not sufficient to forego a safety assessment. Also, depending on the context of use, a GRAS statement is not sufficient to forego a safety assessment. The last internal challenge we encounter, and I would like to bring to your attention, is that FDA-approved flavors and their components can be challenging to identify. This is partly due to inconsistent reporting of flavor names in FDA databases, including those populated by flavor manufacturers. We cannot compare generic flavor names because compositions of such differ greatly, as you've seen earlier, and the quantitative breakdown of approved flavor additives is not listed in FDA databases. Because flavor reviews are very complex, we have created an internal mechanism to support our staff and try to streamline acquisition of relevant data. This allows us to treat similarly situated applicants with more consistency. And with that, we have arrived at our last learning objective. We're going to review some case studies that highlight some of the challenges we discussed today and also showcase how we navigate these complexities. In our first case study, an applicant proposed use of a lemon flavor in a generic diagnostic which is used via the oral route in adults and pediatric patients, birth and older. Unknown to the applicant, the main component of this flavor is propylene glycol. The reference listed drug also contains a lemon flavor, but with a different composition that doesn't contain propylene glycol. No safety justification was submitted by the applicant, and because this product is indicated in pediatric patients, we assess the safety from both non-clinical and clinical perspective. The proposed flavor was not present in any FDA-approved products. Therefore, we needed to employ the approach in which we assess the individual components of this flavor. During an initial assessment, we found that the safety of all components except for propylene glycol could be qualified for safety via prior findings of safety identified in FDA-approved products with a similar context of use. Hence, the only component that's left to assess for safety was propylene glycol. During PharmTax review, we can inform endpoints for which clinical studies are not available by leveraging in vitro and in vivo studies. Our review determined that propylene glycol is non-genotoxic and that no effect levels from published systemic and developmental non-clinical studies determined a large safety margin for the proposed clinical exposure. However, we had to make population-specific considerations. Remember, this product is indicated in neonates and older. Now, propylene glycol is metabolized via the alcohol dehydrogenase to lactic acid and subsequently glucose. Children, especially neonates, have immature alcohol dehydrogenase activity. This renders them at risk for propylene glycol toxicity due to accumulation. The toxic signs are hyperosmolarity, metabolic acidosis, hemolysis, renal failure, central nervous system depression, and cardiovascular decompensation. Other items we identified during our assessment were, for example, that propylene glycol is listed as GRAS by 21 CFR. GRAS are substances added to food that are generally recognized as safe under the conditions of their intended use. We further searched FDA databases and found that at the maximum daily dose of the proposed product, propylene glycol intake did not exceed FDA-approved levels in products with a similar context of use in adults. In neonates, however, we could not find any prior precedents of clinical safety via FDA-approved products. Overall, we concluded that non-clinical data are not suitable to inform safe exposure in neonates. That is because of the difference in metabolic system maturity in non-clinical animal models and neonates. Further, no prior clinical findings of safety for the proposed levels in neonates were identified. Therefore, this is a data gap, and the safety of the proposed flavor was not qualified. Here you see our recommendation to the applicant. And it reads, your proposed flavor contains components that pose an unacceptable safety concern in young infants and neonates for whom your product is labeled. Therefore, we recommend that you reformulate to remove the proposed flavor additive. In this case, we recommended reformulation because non-clinical data are not suitable to address this data gap, and clinical studies are outside the scope of a generic. What this case highlights are two things. For one, the proprietary nature of flavor that's posing an issue for the applicant. Unknown to the applicant, their flavor contained a component that's contraindicated in the patient population. Secondly, same problem, our recommendation had to be written very vaguely because we needed to exert caution to not reveal proprietary information. And lastly, What I want to highlight here is how the context of use matters. Sure, propylene glycol is identified as GRAS, but it still is not qualified for safety in this drug product. Coming to our second case study. Here, an applicant proposed use of a cherry flavor in a generic diagnostic that is used via the oral and the rectal route of administration. The reference listed drug also contains a cherry flavor, but of different composition. The proposed cherry flavor is not present in any FDA-approved products, and therefore we needed to assess the individual components of the proposed flavor rather than the flavor as a whole. Again, no safety justification was submitted. Because this product is indicated in adult patients only, we assessed the safety of this flavor from non-clinical perspective only. As I just mentioned, we needed to assess the flavor based on the individual components, which were 25. And now I'm going to demonstrate the approach we take to alleviate our review burden in such cases. As mentioned several times already, the main principle we apply is a context-driven safety assessment. So because this product is a diagnostic, the duration of use is acute. This means that allowable exposures for genotoxicity and general toxicity can be established based on acute use of the drug product. Next, we looked at the maximum daily exposure to each of the individual components calculated at the respective maximum daily dose of the proposed product. Because the oral and the rectal maximum daily dose differed, we conservatively assessed the safety of the flavor components at a higher maximum daily exposure. Next, we applied our safety threshold determined based on the acute duration of use, which decreased the number of components that needed safety assessment. So we went actually from 20 components down to 5 components that exceeded the appropriate safety threshold. However, keep in mind, as you've seen in the previous slide, the product is indicated for oral and rectal route. Therefore, we task ourselves with assessing the systemic and the mucosal safety of the flavor components. Going forward, we reviewed the safety of the 5 individual components that exceeded our safety threshold by reviewing published literature and information present in FDA internal databases. Our review determined for systemic safety that no effect levels from published non-clinical systemic studies determined sufficient safety margins for the proposed clinical exposure to each of these 5 individual components. The maximum daily exposures to the components also did not exceed FDA-approved levels in oral products. However, for the mucosal safety, we did not identify any published data for any of these components, and only 3 of these components are present in FDA-approved products for the rectal route. Overall, we concluded that the systemic safety of the flavor was successfully qualified. However, there are no non-clinical data to support the mucosal safety and no prior findings of safety to support mucosal safety for 2 of the components. This identified a data gap, and therefore the safety of the proposed flavor was not qualified. Our external recommendation reads as follows. The proposed flavor contains components for which local safety could not be justified based on published information or FDA-approved products with similar context of use. Therefore, provide a justification to address local safety of your proposed flavor. So in this case, we did not recommend reformulation because this applicant actually has a chance to address the data gap with help of non-clinical data. And this case study again highlights how the context of use matters. For one, we successfully applied a safety threshold to decrease the review burden, but we also increased our review burden because now we had 2 main endpoints to assess, systemic safety and mucosal safety. And again, you see how the complexity of these flavor additives and proprietary nature can cause some challenges to applicants and FDA. And here we are at the fun part of my presentation, where I get to test how successfully I delivered my material and how well you paid attention. We have a polling question, and the question is, flavors do not need to be justified for safety in drug products since they are used in foods. Is that A, true, or is that B, false? I'll give you a couple of seconds. The polling should be popping up, and please answer accordingly. And the correct answer is that is false. If you remember from earlier, a crash statement alone is not sufficient to forego a safety assessment. For polling question number 2, I would like to know the following. To support the safety of a flavor in a proposed generic product, an applicant can A. reference FDA-approved products with a similar context of use containing this flavor, or B. submit published clinical and non-clinical information for this flavor, or C. submit published clinical and non-clinical information for the individual components of this flavor, if, of course, the composition is known to the applicant. Or is it D, all of the above? Again, I give you a little bit of time. And the correct answer to question 2 is D, all of the above. A generic applicant has all of these options to qualify a flavor for safety. And with that, I arrived at the end of my presentation. And in summary and conclusion, I would like to highlight a few things again. Flavors are excipients and are reviewed by OGD PharmTox like any other excipients. CFR regulations set the standard for flavors, and FDA guidances recommend how these requirements can be met. We conduct a comparative safety review to the reference listed drug. Safety review of flavors is a risk-benefit evaluation considering the context of use of the proposed product. And depending on the context of use, flavor safety is reviewed by non-clinical and/or clinical disciplines. The safety review of flavors poses many challenges. However, we take actively steps to navigate complexities with flavor reviews. For example, we contributed to FDA guidance describing the information needed for comprehensive safety review. We have ongoing efforts to internally cataloging and leveraging our prior review experiences, and we collaborate with CDER colleagues to harmonize review approaches used for flavor additives. For potential interest, here's a list of resources and guidance documents that we frequently use during our safety reviews of flavor additives. And I would like to conclude by thanking you for your attention, my supervisors, Dr. Shruti King and Robert Dorsum, and the entire PharmTox team of the Office of Generic Drugs. Thank you very much.
Thank you to all of our presenters for their great presentations. We'll now enter into our Q&A panel comprising of our session speakers and introducing Dr. Xiaoming Xu, who serves as the Lab Chief of Branch 3 in the Division of Product Quality Research in the Office of Testing and Research, where he leads multiple research areas including complex formulations, abuse-deterrent formulations, and advanced manufacturing. Dr. Xu is a member of the FDA Technology Task Force and CDER Technology Working Group. As the FDA representative, Dr. Xu also participates in various international collaborations in areas related to nanotechnologies, including standard development and the International Pharmaceutical Regulators Program. We also welcome Dr. Heather Boyce, who works for the Office of Research and Standards in OGD. Heather has over 10 years of experience in the pharmaceutical industry, including expertise in GMP, pharmaceutical product development, bioequivalence trial design and analysis, and generic drug regulation. Heather currently leads the development of product-specific guidance development for immediate release oral drug products. Heather's current topics of interest and research are focused on clinical study design, clinical pharmacology, and generic equivalency of abuse-deterrent formulations. We now have questions coming in. Our first round of questions will be addressed to Dr. Raofi. And here's the first question. For ADF products such as Oxycodone, do we also need to conduct the study with PD endpoints such as taking drug again or the bipolar variables for generic products?
Thank you for that question. A good question. The short answer is no. You only need pharmacodynamic endpoint studies if the RLD includes an aversive agent. And if the generic has the same aversive agent in an amount that's less than the RLD, or the generic has a different aversive agent than the RLD, those are the only situations where you will have to conduct pharmacodynamic studies.
Thank you for responding to that question. We do have a few more questions for Dr. Raofi, and here's the next one. Do the generics have to establish that they are abuse deterrent?
The answer to that is that they have to show that they are no less abuse deterrent than the reference product. In other words, this is a comparison between the generic and the reference, and the generic has to show that it's no less abuse deterrent than the reference for all routes of abuse.
Thank you for responding to that question. I have a few more questions for Dr. Raofi, and here's the next one. How do you select particle size for nasal PK studies?
The particle size should be a particle size that can be insufflated, and that's generally about 1 millimeters or less. And during the development process for the study doses, in the in vitro studies, they have to evaluate the particle sizes, and based on several factors such as drug load, drug loss. This is not a comprehensive list. Based on those factors, the study designers have to choose the particle sizes that would provide the best results. Remember, the point is figuring out to what extent the product has to be manipulated in order to lose its abuse deterrence.
Thank you for responding to that question. Do have another question for Dr. Raofi, and this is it. Is a control IR product needed in comparative nasal PK studies for generics?
The answer is no. In a sense, the RLD is your control in this situation and your comparator. Although controls are needed for ingestion and injection for nasal PK studies, an IR control is not needed.
Thank you for responding to that question. We have one more question for Dr. Raofi in this round, and here's the question. Why is it important to characterize the manipulated product? In real world, the abuser will grind the tablet and snort all of it.
Because basically when you're comparing generic to the brand in the context of ANDAs, you have an additional burden of comparing the two products. And that's why you need to carefully characterize so that you know exactly what you're comparing.
Thank you for responding to those questions. We'll now move on to Dr. Al-Ghabeish. And here's the first question. In slide 13, I noticed that none of the RLDs have an abusive deterrent label for the inhalation route. Does this mean that generic applicants do not need to test for this route?
No, they actually still need to test for this route. As I mentioned in my talk, the generic applicant has to show that their product is no less abuse deterrent than the RLD, in all routes of abuse. We still, the generic needs to show that they will not have an additional risk than the RLD in all the routes.
Thank you for responding to that question. We have another question for Dr. Al-Ghabeish, and here it is. In slide 20, can you please elaborate more about the effect on the number of tablets used, milling time, drug loading on milling efficiency?
I'm going to try to make it brief, but in that study, and specifically to that specific product and the API or the model drug that's been used, the milling efficiency is related to the fine particles produced, the percent of the fine particles produced. So it was shown that the number, as you increase the number of tablets and increase the milling time, the fine particles percentage increased, which meaning that the milling efficiency was higher. For the drug loading, that was a bit tricky because the drug was mainly composed, the product was mainly composed of the drug in the AD polymer. So increasing the drug loading reduced the AD polymer percent. So actually, the increase in drug loading generally caused increase in the milling efficiency because there was more drug and there was less AD polymers. This was kind of a bit tricky for that specific product. Also, what's shown in that particular study also that when milling the product, the percent, the milled product has a different percentage of the drug, which could be different from different products. And that was an interesting observation.
Thank you for responding to that question. We've got a couple more questions for Dr. Al-Ghabeish, and here's the next question. If the proposed generic product showed to be superior to the RLD in some of the abuse-deterrent evaluation studies, but failed in one evaluation, can it still be approved?
The short answer will be no. The thing, as we mentioned many times, the generic has not to show more risk than RLD for abuse. So even if it showed to be superior in all different routes, if it failed that one route, that would increase the risk to be abused in that route and could be a more even dangerous route. But I also said before, just not approving that product, we all usually ask the applicant to look back to the methods. Sometimes that the failure may not be that the product itself is failed, but it could be the method picked. It was not an effective method. It could have not a robust, it has so high variability, so that the difference was not a true difference. And also, as I mentioned in the article, in the presentation, before even designing the product, it's very important to look for the product quality attributes before designing an IDF generic. Thank you.
Thank you for responding to that question. We have one more question this round for Dr. Al-Ghabeish, and here's the question. You mentioned drug delivery as one of the AD designs. Can you please explain how drug delivery can be in an AD design?
Yes, I have mentioned that in the slide. Sorry, I did not have the time to explain that. Drug delivery could be that if you picked up a method of delivery, or if you picked up a release design that by itself would actually even be resistant to abuse. For example, if you do a subcutaneous implant or you do an injectable depot, that is hard to actually get out of the system and extract the drug. So that by itself, that drug delivery system by itself could be resistant to abuse.
Thank you for responding to that question. We'll now move on to Dr. Wu for the next few questions. And here's the first question. What factors, if any, in PBPK model potentially explain observed food effect?
Thank you for that question. The food effect is a complicated process. The impacting factor can include delayed gastric emptying, the increase of secretion of bile salt, and increase of viscosity of GI fluid. So in vitro data such as biorelevant or biopredictive dissolution data to mimic this situation for different formulations and put into PBPK model can help to evaluate the risk of different bioavailability attributable to food intake. In addition, the physiological parameters that can reflect these changes under fat condition can also help explain the possible observed food effect. Thank you.
Thank you for responding to that question. We have another question for Dr. Wu, and here's the question. Can we predict the impact of incipients on the dissolution absorption of the main API drug by PBPK M&S?
Thank you for the question. We can conduct a sensitivity analysis on the critical parameters associated with excipient impact in PBPK modeling. For example, the permeability to predict the impact of excipients on the drug absorption or drug exposure. Our FDA colleagues have published a paper in this regard, and also it is important to conduct in vitro experiments using biorelevant conditions to evaluate the impact of excipients on dissolution or permeation. Thank you.
Thank you for responding to that question. We'll move on to Dr. Mueller. I have a few questions that came in for Dr. Mueller. And here's the first question. Is it mandatory to justify the levels of flavor and its components if the entire flavor constitutes less than 0.1% weight of the total formulation?
That's a great question. And actually, I think I saw the question come in before I touched that point during my presentation. So in short, as I mentioned, the amount of a flavor as a percent of the total formulation is not sufficient to forego a safety assessment. And that is simply because 0.1% might not seem a lot, but if you look at the maximum daily dose of an oral formulation, exposure to the flavor or its individual components likely exceed safety thresholds, and therefore the safety still needs to be assessed. The flavor still needs to be qualified, even if it's less than 0.1% of the formulation. Thank you.
Thank you for responding to that question. The next question for Dr. Mueller. Quite often, all constituents of the flavor may not be listed in the AID database. Please share your thoughts on how we should go about, a generic potential applicant should go about justifying such components of a flavor.
Yeah, I certainly agree that is a problem, and that is a problem for us on FDA side as well. It's simply due to the proprietary nature of the flavor additive. One recommendation I have is to submit a controlled correspondence indicating what flavor you intend of using. However, we still would need to have access to the individual components to conduct our controlled correspondence assessment. And that's simply a check if the flavor, if there's anything known about its flavor or its components in FDA-approved products.
Thank you for responding to that question. We have another question for Dr. Mueller. And here's the question. Is there a list of components that are not recommended for use in pediatrics? And you gave the example of propylene glycol as being an ingredient for concern in pediatric patients. Is there a reference that lists other known components of concerns for pediatrics or other vulnerable patient populations?
That's a great question, and you're not the first person to ask me these questions, and we even get this question internally. The answer is no, there is not a comprehensive list of components that are contraindicated in pediatrics or in vulnerable patient populations. There's certainly some components that have known safety signals or where you would think twice, such as the propylene glycol. Ethanol is another one, benzyl alcohol, for example. However, even components that are seemingly harmless in any given context of use, they might cause some increased adverse events, or they might change the safety profile compared to that of the reference listed drug. And one simple example is, medium chain triglycerides. You have them in a large amount in an antibiotic that's causing diarrhea as a side effect, and now you have a solid amount of medium chain triglycerides in that same product. That might increase the risk of the side effect. And if the ILD doesn't have that, we cannot be changing the safety profile, and that is not allowable or acceptable from our perspective. So basically, any ingredient under any given context of use could cause an issue. And so, no, there is no comprehensive list. I hope that explains why we don't have such lists and keep such lists.
Thank you for responding to that question. We received a question directed to Dr. Hsu, and here is the question. Based on the two speakers' talk about abuse-deterrent formulations, it looks like it is not foolproof to prevent people from actually abusing the product. What is the point of conducting research on something that does not work?
Thank you for the question. Why do we do research? This is indeed one of the dilemma and also the challenges, the biggest challenge facing us in the regulatory science development for the abuse-deterrent formulations. As Manar mentioned in her talk, that we have to keep in mind a very delicate balance between reducing the risk to the abuse for the abuser population, while ensuring patient access. So from that perspective, the driver of FDA's research in this area is really to enhance the affordability of the medicine to the patient, because as we know, since 2011, the opiate, especially extended dosage form, this category has been dominated by the brand name drugs, and we need access to the generics to bring down the cost. Now, why do we need to do research? As with any new scientific endeavors, there's generally a lack of understanding at the very beginning. And this is especially true for the abuse-deterrent formulation. And the example shown by Manar and Saeid showed a lot of progress have been made in this area to improve the understanding. And because of those research, we were able to develop recommendations in the guidances, like the general guidance as well as the product-specific guidance, as well as to support the assessment in the and application reviews that include the first approved generic in this category. That's why we do research. Thank you.
Thank you, Dr. Hsu, for responding to that question. It looks like we have just under 4 minutes remaining in our Q&A panel session. And we had a question just come in for Dr. Boyce. So the next question will be for Dr. Boyce, and here's the question. Can the particle size data presented in Saeid's presentation be used to support a waiver request for in vivo comparative nasal insufflation study recommended for the ADF products that contain polyethylene oxide?
Hi, that's an excellent question. I would say at this stage, it's probably too premature to be able to use just purely particle size data to predict PK results. As Saeid presented, there are other factors that could be important, such as the excipient to drug ratio. In the presentation Saeid presented, this was not a significant factor that influenced PK variability. However, the study was limited to very specific range that was studied. So, from a generic perspective, that range could be larger. Also, there are other factors such as mucoadhesive clearance that may need to be considered as well in these type of predictions, and as well as dissolution data would be important. So this type of work is ongoing in ORS, but at this stage, it's just using the data by itself would not be suitable to request a waiver for insufflation studies.
Thank you for responding to that question. It looks like we have time for just one more question, and we'll address the final question came in for Dr. Mueller, and here's the question. If the agency issues a comment to reformulate due to a safety concern with the flavor composition, does that mean that a safety assessment route is ruled out?
That's a great question. Thank you. Yes, so typically if you get the recommendation to reformulate, we have done our due diligence, and there is likely safety assessment conducting studies is likely either not in the realm of a generic or it's not feasible. And again, we have to be somewhat vague to not give away proprietary information in these cases, and I understand how that can be frustrating to industry. There's always a way of issuing a general correspondence to inquire more about our recommendation, but typically, yes, it means the data that would need to be submitted are not in the scope of a generic. Thank you.
Thank you for responding to that questions, and thanks to all of our Q&A panel for responding to numerous questions that came in, and our panelists that also joined us. We'll now transition into our lunch time and return